Hello dear colleagues, my name is Jan-Niklas Eckardt. I'm a hematologist and medical oncologist at the University Hospital in Dresden, Germany, and I lead a research group developing AI-based tools for diagnosis and management of hematological diseases. Today it is my pleasure to introduce you to synthetic data for cancer research. We'll explore what synthetic data is in the first place, why synthetic data may help us overcome a variety of hurdles that are currently troubling healthcare systems and clinical research, and I want to introduce a few use cases for synthetic data in hematology given a few of our recent publications. Alright, let's get into it. First, a quick glance at my disclosures and potential conflicts of interest, none of which bear any relation to this presentation. Okay, so we should start with examining what we talk about when we say synthetic data. And let me give you an example. As a 90s kid, to me at least, there's only one real Spider-Man, Tobey Maguire, of course. But recently, other Spider-Man movies have come out starring other actors than good old Tobey over here. One might say we have an original Spider-Man and a couple of imitations that mimic exactly what the original does. They look alike, they wear a very similar suit, they have the same role, the same powers, so for all intents and purposes, these imitations are more or less indistinguishable from the original. This analogy, in essence, captures what synthetic data are. They are based on real data, anything from text, images, tables, sounds, and they are generated using feature distributions in the real data, and they closely mimic the behavior and the look of the real original data. Features in our example could be Tobey's hair color, the Spider-Man suit, or Spider-Man's superpowers. In a medical context, these features could be visual, for example, the morphology of cells under the microscope, or could be probability distributions of certain genetic alterations in tabular data. So the bottom line is synthetic data are generated by training models to replicate feature distributions of real data, and then they closely resemble real data to the point where they ideally become indistinguishable. Okay, so now that we have an understanding of what synthetic data are, basically the question remains, what are they good for? First of all, data availability and data sharing, especially when it comes to publicly available medical data, is a big issue in healthcare research. Oftentimes, privacy concerns are referenced when it comes to sharing data or making data publicly available. And don't get me wrong, privacy is super important, and blindly sharing medical data is of course very dangerous. But a recent study by the World Economic Forum suggests that at least 97% of data generated in clinical routine around the globe goes completely unused. Just imagine what we could learn about the biology and epidemiology of the diseases we treat if we intelligently use a bit more of that data. Sharing data between centers, even within the same country, is often a rather bureaucratic process. Again, patient privacy is the big hurdle here. Further, data generation, as all of you may know, is a very cumbersome process. Experiments and clinical trials take time and are increasingly costly. Also, data diversity is a big issue. Most patients in clinical trials, at least in Western nations, are not really diverse. So can we actually infer all our findings from clinical research to other patient populations? As you can see, there are a lot of issues currently troubling clinical research. Let's explore how synthetic data may help that. Before we get too deep into use cases, I'd like to introduce only a bit of the technology behind synthetic data generation. Of course, the idea of synthetic data generation is not necessarily new. There are numerous statistical approaches that have been developed over the last decades, but the whole field got catapulted forward by the introduction of artificial intelligence-based models, most prominently generative adversarial nets, or GANs for short, introduced by Ian Goodfellow and colleagues in 2014. To understand GANs, this sentence from the abstract is key. A generative model captures the data distribution, and a discriminative model estimates the probability that a sample came from the training data rather than the generative model. Let's see how that plays out. Okay, so let's assume we want to train a GAN model to generate synthetic image data from, for example, bone marrow smears. As we have learned, the GAN itself is composed of two networks, the generator and the discriminator. The generator uses a vector of random latent variables as an input. You can basically just think of that as noise. Based on that, the generator generates synthetic data. The discriminator then has the task to evaluate whether any input given to it stems from the generator or from a real data set, in our examples, bone marrow smears. Based on the accuracy of the discriminator's judgment, both networks are updated. Over time, this leads to the generator being able to generate more and more realistic data. The GAN converges when the discriminator is no longer able to distinguish between which input is real and which input is fake. So essentially, you can think of this as a digital game of cat and mouse. Here's an example of what this might look like. I want you to take over the role of the discriminator now. What we did here was we trained a GAN model to generate synthetic image data from bone marrow smears. And of the images you see here, the question is, which do you think are real and which do you think are fake? I'll give you a couple of seconds to make your guesses, or you can pause the video if you want to really thoroughly go through these images. Okay. There you go. Don't worry if you didn't find all the mistakes. We showed these images to multiple hematologists, and the overall accuracy of finding the fakes was only around 60%, meaning that even seasoned veterans in cytomorphology were not really able to reliably tell synthetic and real images apart. Okay, but what is that good for, you may ask? I know this is a super busy slide and I apologize, but let's dissect it real quick. One of our key areas of interest in the lab is to develop computer vision models that diagnose different hematological malignancies based on microscopic image data. We've previously shown that AI can reliably distinguish between AML, APL, and healthy bone marrow donors. Now for this experiment, we trained multiple image classification models with varying proportions of real and synthetic data. So we start by training model A to classify, for example, AML versus donors with 100% real data in the training set, measure the accuracy in the area under the curve, and then we randomly remove 10% of the real data from the training set and substitute it with 10% synthetic data, like the ones that you've seen before. We measure the performance again and so on and so forth until we reach a fully synthetic training set without any real images in there. As you can see across the board, all use cases, the performance is remarkably high, even with almost no real training data of fully synthetic training data sets. So the TLDR here is that real image data and high-quality synthetic images of bone marrow smears can be used interchangeably to train computer vision models for the diagnosis of hematological malignancies using microscopy images. And I think that's a big deal. So at least in my opinion, this means that we can effectively generate synthetic image data from any real microscopy images as long as image quality is sufficient. And then you can use these data to train image-based classifiers or eventually boost the performance of existing classifiers, or you can even validate the performance of external classifiers developed by a third party. Images, of course, are a nice way to visualize things, and I hope you caught the general idea of how to generate synthetic data and how to use them. But now it gets really interesting. We thought if we can do that with images, maybe we can do that with, let's say, whole patients as well. And by quote-unquote whole patients, I mean comprehensive patient-level information. So for example, demographics, clinical data, laboratory test results, genetics, so on and so forth. Thinking about clinical trials, one big issue, of course, is recruiting enough patients for a clinical trial is hard. Recruitment failure, in fact, is one of the top reasons for high failure rates of clinical trials, especially in oncology. What's more is that given the ongoing sequestration of entities within our field, so let's say we don't only talk about NSCLC, but we talk about ALK-positive NSCLC and so on and so forth, it's going to be even harder to recruit patients into trials as more and more targeted therapies emerge. As you are aware, clinical trials cost exceedingly more and take longer and longer until completion. So the issues we know exist for real patients can essentially be overcome by generating synthetic patients with, for example, generative artificial intelligence. For synthetic patients, privacy is not an issue. They are not real patients, so they can simply be shared. They just mimic the behavior of their training samples. They are adaptable, so for instance, you can either force models to generate samples with certain attributes, let's say height or specific genetic alterations that you're interested in, or you can generate them at random and then later on only select those with features that you desire. They are scalable, meaning as soon as you have a trained model, it doesn't really make a difference whether you create 1 or 1 million synthetic patients, and it's super cheap and fast. So with a trained model, only with the click of a button, you can basically generate a large cohort that you can, for example, use as a control cohort in a prospective trial. So that's just what we did. We took data from 4 previously conducted trials of the German study Lions leukemia, using acute myeloid leukemia as an example disease with a patient number of 1,600 in the original training cohort. We used 2 different methods to generate identical numbers of synthetic patients. One is CTEP-GAN+, a generative adversarial network, very similar to the one we've talked about before. And the other one was normalizing flows, which is not a GAN, but for time's sake, I'm not going into too much of the technological details here. So what you can see here is the frequencies of a multitude of molecular and cytogenetic alterations related to myeloid neoplasms that were available for the original training cohort. So the real patients here are depicted in the fully colored bars. And the corresponding distributions for generated synthetic cohorts, so CTEP-GAN+ in stripes and ENFLOW in little dots, you can see on the plots as well. We see here that both synthetic cohorts closely mimic the frequencies of alterations found in the original cohort. There are a few dissimilarities, of course, but nothing completely out of the ordinary. For example, if you were to compare distributions between different study groups, you might see something like this as well. Also bear in mind that we compare 1,600 real patients versus 1,600 synthetic patients from one network and 1,600 synthetic patients from another model. So given these numbers, even minuscule differences turn out to be statistically significant. And what's more, we tested the intervariable relationships between all of these variables you can see here and outcome. So complete remission rate, event-free, relapse-free, and overall survival. We found that all of the effects that were found in the original cohort, so for example, NPM1 mutations are prognostically favorable, TP53 mutations are associated with a dismal prognosis. All of these effects were preserved both in their directionality and in their significance level in the synthetic cohorts. At the same time, none of the patients from the original cohort could be re-identified using the synthetic cohorts. That means study groups and/or hospitals could essentially generate synthetic data from their patient cohorts, and others could explore these synthetic data sets, for example, to find new risk markers or biological subgroups without compromising patient privacy. Next, we evaluated patient survival, comparing the original cohort in blue with both synthetic cohorts in red and yellow. And here we can see an interesting behavior. Generally, the curves in both Kaplan-Meier graphs are somewhat similar. However, we do not see a plateau at the end of the follow-up period for the synthetic cohort generated by CTEP-GAN+ in red, and ENFLOW in yellow tends to overtly censor patients at the end of the follow-up period, as you can see in the risk tables below. And honestly, for now, I do not have a good explanation for this. One thing that comes to mind is that the models we used are not designed to deal with time-to-event data, and the long follow-up period also may be an issue. But there are a few models out there, like for example Survival-GAN, that are explicitly designed for use cases such as this, and we tested them, but found its performance to be way worse than what we see here. So judging by the log-rank p-values that you can see, there are significant differences between original and synthetic data when it comes to time-to-event outcome data, with the exception of overall survival for the ENFLOW generated cohort. And I believe such a cohort could, for example, be used as an external synthetic control cohort in prospective clinical trial designs. But of course, model optimization is still needed. So speaking of optimization, we recently put together a technological toolbox to train, test, and evaluate synthetic data generation models. And as you have seen, not every model is alike, and some models may be better suited for given use cases than others. The most important thing is that you know what you're looking for. Your synthetic data fidelity, meaning how well do your synths resemble the real training data, and its usability, meaning how useful are your synthetic data in the context of what you actually want to do with them, for example, survival analysis. Making sure you train and optimize your models using the right tools is very essential from our experience. But finally, I want you to be aware that synthetic data may help with all of these issues that we discussed before and may actually make an impact for data sharing and clinical trial designs in the future. But off the shelf, they are no silver bullet. AI is rarely a magic lamp, and both researchers and clinicians should neither under nor overestimate its capabilities in the healthcare sector. A few examples of how synthetic data may cause harm are listed here on the iceberg. For instance, if your training set is very homogeneous or biased, These biases may be carried over by generative models to the synthetic data set. Now imagine that at some point this synthetic cohort may be used in the context of a prospective clinical trial as a control cohort for a drug to seek approval. This may cause actual harm to minority groups that are not represented in the synthetic data. And further, I mentioned that synthetic data preserve patient privacy, and at least in the examples from our research that I've given, that's true. But we also took countless measures to make sure that data cannot leak and that the synthetic models are bulletproof to re-identification attacks, at least as bulletproof as you can get them. But if an adversarial third party were to get access to the fully trained models and at least partially to the full patient data from the training set, re-identification attacks could be successful. So when you train your models, you have to be aware of the fact that off-the-shelf generative models do not guarantee privacy by default. Many models allow you to add noise to the data or define a so-called privacy budget, but this is a delicate balance between privacy and usability. You still want your synthetic data to resemble the original data as closely as possible, but you don't want to jeopardize patient privacy. You have to find a balance between the two. Also, when it comes to generating synthetic data at scale or selecting for certain attributes in the context of clinical trials, adversarial actors could basically generate synthetic data that supports their hypothesis. And in order to prevent this cherry picking, I think we need more regulatory oversight and ideally a party that generates the synthetic data that is separate from the research group, for example, from a drug company that wants to issue a drug filing. So in conclusion, I've given you a brief overview of what synthetic data are, how they are generated and what they can be used for, and also how they can be misused in the context of healthcare research. If you're interested in what our research team at TU Dresden is up to apart from synthetic data generation, you can visit us at aincancer.org or scan the QR code. And I want to thank my amazing team and of course our amazing collaborators and sponsors, without whom I probably won't be standing here today. And finally, I want to thank you very much for your attention and I hope you have a great day. Thanks.